My name is Daniel, and today I'm going to present part of my PhD studies, and I've titled it Investigating Grain Filling Duration for Improved Yield in Grain Sorghum, and my supervisory team is listed down there. So many of us are familiar with this diagram, and this was borrowed from Mark Cooper. So just to, when you look at that diagram, I'm working on this nexus here that you can see. That is a grain number, grain size, and its contribution to yield. And I've bolded it here just to illustrate a very simple way, in that if we have a relationship between grain number and grain size, and if you multiply grain number by grain size, that gives you yield in many of the crops that we work with. And for sorghum, that is not any different. This is very simple, and you can appreciate from this crop growth model, genotype to phenotype trait model, there is a complex number of genes that are controlling different developmental processes, together with some environmental cues that all interact to either give you changes in grain number, grain size that will ultimately lead to yield. So I'm just presenting this in a very simplistic way. And we saw some presentation recently by Daniel, and he showed this diagram, which I also borrowed, and just to illustrate, there's a trade-off in many crops, especially for grain number and grain size. And this diagram just shows the selection response under different trade-offs, in that if you increase your grain numbers, chances are that you are decreasing your grain size. So you may get a genotype with higher numbers, but the amount of size of those grains will be smaller. And this has been exploited mainly by breeders for yield improvement, and sorghum is no different. Most of the changes in yield in sorghum have been achieved through increases in grain number rather than grain size. However, there is an opportunity to increase yield through both increases in size and in number. And I will illustrate that on here. In the graph that you see there, the y-axis shows the 1,000 kernel weight, which is a proxy for grain size, and the x-axis is the grain number. And in the purple dots, it's the relationship between grain size and grain number in high yield environments, and there is no stress in that environment. And in the other red dots, it's in low yield and high stress environment. But still, we can appreciate that that trend is still there. However, as you can see, there is a nexus for improving that. There's a deviation for grain size for a given grain number. So this can be exploited if we mine the native variation that is available for grain size in sorghum. And this we've shown in Australian breeding programs, both in test and released hybrids. And in this diagram, I just show that there is a general relationship between yield and grain number, and there is a positive contribution of yield, of grain number to yield. However, in hybrid combinations, as you can see here in the lower panel, whereby hybrid grain number almost always is bigger than the grain number for the inbred parents that are used in many of the breeding programs. So this scenario is replicated in either the released hybrids or even in the test hybrids that are there. But for grain size, if you see here, the grain weight for the hybrids and the grain weight for their parents, they are sort of fall on the one-to-one line, showing that there is very minimum contribution of grain size in the parental pool into the hybrid pool. So there's it's a bit constrained, and this constraint is mainly through this strict selection that has been done for the sorghum hybrids. And the next image will just show us how this constrained diversity in the sorghum parental pool is. This PCA diagram shows the entirety of the sorghum gene pool that is available, and the colored sections in blue is the male parents that contribute to the hybrids that are commercially available, and the pink portion shows the female pool. So for all the hybrids that are being developed currently, or the commercialized hybrids, most of their variation is just sold from this section that you can see here. However, all the gray sections that you see has been exploited to a very little extent, and that is for valid reasons, because the hybrid breeding system is limited by the cytoplasmic male sterility that is in the female genetic pool. So we sought to look at what are the components that contribute to grain size, and how can we explore these components further to try and improve yields in sorghum through manipulation of the grain sizes. And here you can just say that the genetic potential for maximum grain size can either be contributed by the grain filling duration, and grain filling duration is the period between flowering and physiological maturity in sorghum, or in most of the cereal and legume crops. And also there is this, the rate of the grain filling, and this is how fast the assimilates are packed up into the seed. And then we have another component, which is the assimilate availability. That is the availability of carbon from the source into the sink. And this assimilate availability, in many cases, modifies both the rate and the duration of grain filling. So to answer these questions, we sought to explore the most of the available genetic diversity for sorghum, and this PCA is just like the previous PCA, but this one now shows all the racial groups that are available in sorghum, and these corners represent the different racial groups. And for the sorghum hybrids that are available commercially, they have been developed mainly using these two races, mainly the Kaffir and the Caudatum. However, the Guineas and the Duras have been used to a very little extent in commercial hybrids, and we sought to look at this entire diversity to understand the extent of this trait variation for the traits of interest. So we have a high diversity of around 1,000 genotypes, and these genotypes have some genetic data as well, and it's a good point of entry to explore the native variation for any traits of interest. So we had 4 main questions that we asked, like, can we improve grain yield in sorghum by modifying the grain filling duration? And from literature, there are many ways that have been recorded to estimate grain filling. And we sought to seek if we can develop a high throughput method to estimate grain filling duration, understanding that if we measure a trait well enough, then we can have the latitude to improve that trait, knowing the variation or the exploitable variation that is available. We also sought to understand the physiology behind these traits, and also try and predict the putative benefits of these traits in production environments of interest for sorghum. And the last aim was to identify any genomic regions that underlie grain filling duration. However, in the interest of time, I'll discuss the first 3 questions that we sought to answer in this today's talk. So in breeding, we say if you can measure it, chances are that you can improve it if you know the limits and the available genetic variation for a trait. And to develop the methodology for measuring this trait. We looked at the literature and found that some aspects that could modify grain filling, in many studies, they've not considered this either environmental or other factors. And I will just list them and then show how we looked at them in our measurements. So in that image, you see the first time this is a floret, flowering. And then the next image just shows the progression of developing grain from when the grain filling starts and then until we get this black layer, which is an indication of attaining physiological maturity. So previous methods that have measured grain filling duration have not accounted for within or between plant variation. Many studies have not accounted for temperature effects, especially on flowering, because plant growth responds to changes in temperature, and it is important to look at the temperature variation. Another thing is the relative position of the seed or the grain that you sample in the whole panicle. And then panicle position relative to the position of the sun. And here we call it heat load, and I'll explain later why it is important. And also we looked at assimilate availability, that is supply of the assimilates or the strength of the source relative to the sink. So to account for plant variation, we just selected individual plants within our experiment where we measured both the flowering and the grain filling duration while accounting for the effects of temperature. For the 3 experiments we had one at Gatton, one at Hermitage, 2 at Hermitage in 2 different years. And when we estimated grain filling duration in days, the histogram on the left just shows that in Gatton, where the planting was done late and flowering occurred towards winter, if you estimated grain filling duration in days, you would see that there is a big difference between the distribution of the grain filling duration for these common genotypes, both in Gatton and as compared to both Hermitage and Hermitage 1 and Hermitage 2. However, when we estimated this grain filling duration in terms of thermal time or degree days, we find that Gatton especially comes together with the other 2 sites in terms of similarities. And if you look at the genetic correlations across the 3 environments, we found that especially for correlations between Gatton and Hermitage, it really improved from 0.3 to 0.5, showing that accounting for thermal time improves our estimation for grain filling duration. Another thing we looked at is the panicle position, and this was important because flowering in sorghum takes an average of 5 days from flowering from the tip to the base of the panicle. And here you can appreciate that in this panicle, the top panicle, part of the panicle has already completed blooming. The middle part is just flowering, and the basal part has not yet started flowering. So in many of the previous studies, they estimated flowering at 50% flowering at of the panicle, but grain filling duration is estimated at the base of the panicle. So we just sought to investigate if estimating grain filling duration at either of these panicle positions would be representative of the entire panicle. And to do that, we identified a panicle position, and then when that panicle position started flowering, we recorded the flowering, and again, at grain filling duration, at the end of physiological maturity, we also measured physiological maturity at those respective positions of the panicle. So we ended up with 5 panicle positions, and each of these positions, we measured both flowering time and grain filling duration, just to understand if that variation is across the panicle or within the panicle. We also report now that across the environments that we did this experiment, we found that there was little variation in the grain filling duration across the panicle position, that is on the y-axis, and grain filling duration in days on the y-axis, and panicle position on the x-axis. For the first experiment, there was only significant differences between panicle 1 and 5 and panicle 2 and 5. In the second experiment, there was no significant difference between the different panicle positions. So we investigated this further and looked at the different correlations between these panicle positions. And the lower panel here, the off diagonal, just report the correlations across the panicle positions for grain filling duration in one experiment. And the diagonals are the repeatability for all these measures across the experiment. So we can appreciate that across the different experiment, there was very high repeatability across the panicle position. And this is important for breeders because we are more interested in the ranking of these genotypes across environments. And this shows us that measuring grain filling duration at one panicle position, when we are consistent, then that panicle position can be a representative of the whole panicle. And in this case, we suggest that measuring grain filling duration at the top of the panicle is more repeatable, and it is easier to measure at scale. The other point we looked at was the heat load, and this is noted because for any panicle in the field, we noticed that one side of the panicle was maturing at a faster rate than the other panicle, and we set up some thermal cameras in the field, and we noticed that in the side of the panicle facing the sun was always warmer or hotter than the shaded part, up to 5 degrees hotter. And when we selected different genotypes across the experiment, we showed that across the whole experiment, the shaded part took longer to achieve grain filling duration than the sunny side. And if we look at this regression of the shade and the sun side, all the genotypes except one either had the same grain filling duration across the sunny side, or the shaded side was always taking longer to attain grain filling duration than the shade side. When we looked at the assimilate availability, and this treatment was imposed at flowering, where one panicle was chopped into half to reduce the number of seeds and increase the assimilate availability for those seeds, we show that there was no difference between genotypes that were in the half head or the full head. So assimilate availability did not modify grain filling. And we show that the full head and the half head had could explain up to 50% of the observed variation in this grain filling duration in either full or half head. So we looked further beyond the grain filling duration and looked at the grain filling rate. And the results show us that for the half head, there was an accelerated grain filling rate. And that's how the half head achieved bigger grains than the full head. And this, we think, is because the half head attained maximum grain filling rate faster and then was sustained for a longer period. So with these results that we got, we sought to investigate further the physiology and the genetic variation for grain filling duration. And we show that the grain filling duration across the diversity panel that we observed had medium heritability of between 41 and 61%. And this shows that this trait can be improved through breeding. We also looked across the different sorghum races, and we show that here there is almost a constant mean for grain filling duration across most of the races. However, for the Guinea race, in all the circumstances and all the environments that we tested, the Guinea race almost always had a higher mean for grain filling duration and had genotypes with longer filling duration than all the races. The Guinea race in sorghum has not been used in the sorghum breeding program yet. We also looked at the correlations and the association between grain filling duration and other yield components like the 1,000 kernel weight, that is a surrogate for grain size. And we saw that there is a positive contribution of grain filling duration to 1,000 kernel weight. We also looked at grain number per panicle. We found there was a negative significant correlation between grain filling duration and grain number. In some cases, no significant differences, but it was very, very small, meaning we could improve grain filling without penalty to the grain numbers. We also looked at flowering time, and we show that here we could have genotypes that are earlier flowering, but still have longer duration and vice versa, meaning we could improve grain filling or modify grain filling without penalties to the flowering time. And lastly, we looked at the plant height, which is an important trait for many hybrid breeding programs. They have a limit for this plant height because of commercial large-scale mechanized harvesting. And we saw that there is a positive correlation, yes, with flowering, with plant height, but we can modify grain filling duration without penalties to plant height. So the next question we asked is if there is any exploitable variation in grain filling duration, and could we use it to modify grain sizes? And here the y-axis shows the 1,000 kernel weight, which is a surrogate for grain size, and the x-axis shows the grain filling duration. The point on the graph there is the point where a successful commercial line sits, and we tend to exploit that part of the graph, meaning if grain filling duration is a trait that is controlled by many genes with small effects, then transgressive segregation is possible. And we try to answer this question through a modeling platform where we attempted to predict the impact of increasing grain filling duration and how that will have an impact on yield across Australian target population of environments where sorghum is grown. And we simulated some genetic parameters where we simulated a standard genotype that is Buster and then added 10% grain filling duration or 20% grain filling duration, and also simulated a reduction in flowering time with an increase in grain filling duration. And this was done across 3 locations using 60 years of data and standard agronomy. We also tested this across the different environment types that are possible. And this just shows supply-demand ratio for water. And this is centered around flowering. And in the first environment type, it shows water is non-limiting across the whole season. Second environment type shows at flowering, water becomes limiting. And environment 3 shows there is a limitation of water from the beginning of the season, and then after flowering, that limitation is relieved. Number 4 and 5 shows a gradual decrease in water availability and then no relief of that water stress. So this comes up with these 5 possible environment types. And we show that for this standard genotype across the different environment types, we had an increase in yield when grain filling duration is increased by 10%, an increase in yield of between 3 to 8%, and when it is increased by 20%, of between 6 to 14%. But the more important thing here is in environments that water was non-limiting, we got a big increase. That is environment 1, 2, and 3. And environment 4 and 5, where water was more limiting post-anthesis, we found there was a marginal increase in yield. So when we look across all the tested environments, that is 3 environments in Dalby, Emerald, and Tamworth, graph number A shows when grain filling duration is increased by 10%. And then number C shows when grain filling duration is increased by 10% and when flowering time is reduced. And here we can appreciate that when we increase grain filling duration by 10%, majority of the benefits come from the environment types that are not water limiting. Here, the darker symbols shows environments that are not water limiting, and the open symbols here shows environments that are limited in water, especially post-anthesis. However, if we reduce the flowering time while increasing grain filling duration, majority of these benefits of grain filling duration is in those environments where water is limiting early and then non-limiting post-anthesis. So in environment 4 and 5, if we reduce flowering time, then we get some increases benefits of grain filling duration. And this is just because the water is conserved early and that water can be used later in the season. So to summarize, we established that the position on the panicle basically has no effect on the estimated grain filling duration, provided the sampling is consistent. If we are sampling consistently from the primary spikelets, then we don't get any differences of grain filling duration estimated across the panicle. That accounting for thermal time and heat load improved the estimation of grain filling duration. We found that assimilate availability had relatively small effects or no effects on the estimated grain filling duration. And again, the assimilate availability had more effect on the grain filling rate rather than the grain filling duration. And there is exploitable variation for grain filling duration in these exotic genotypes beyond what is available in the current commercials. And we also argue that if grain filling duration is under complex genetic control, then we can improve and get genotypes that are having grain filling duration beyond the range that we've observed. And what we are seeing in this study, as much as we found that grain filling duration has the potential to increase yield, there are some other considerations that are possible that we've not looked at. For example, if we increase the yield through grain filling duration, what are the other negative things that will come with it? For example, if we increase yield, sorghum will tend to lodge, and we did not simulate any potential negative effects of increasing yield and getting lodging. Another thing which is important for many production systems is how these longer filling genotypes will fit into the current cropping systems, especially for growers that are doing multiple cropping. If grain filling longer genotypes will stay longer in the field, it means it may need to mine the available water resources, especially in the lower soil levels. And if this is not fitting in the cropping systems of whatever the farming systems the growers are using, then potentially that will be a negative contribution of this grain filling duration to the cropping systems. And that said, then we acknowledge that grain filling duration is potentially a useful trait that we need to explore more to understand its genetic control. Thank you. I would welcome any questions. I thank our Hermitage sorghum research team and all the contributors to this work. Thank you.